Так, мне кажется, я разобралась. Сейчас включим слайд. Okay, the topic of today's lecture will be radionuclide therapy, and radionuclide therapy is a very multidisciplinary subject that we have a collaboration of specialists on the preclinical side that are developing the radionuclide-based pharmaceuticals, and then we have specialists on the clinical side that are using it for applications. And it is a multidisciplinary area because you need to think about many different disciplines if you are developing a radiopharmaceutical. For example, you need to consider chemistry, radiochemistry, biochemistry, biology of your target. You need to consider the production of the protein using biotechnology. You also need to consider radiopharmaceutical aspects, pharmaceutical aspects, preparation, quality control. And then on the clinical side, it's usually a collaboration between physicists who are doing imaging work, radiochemists who would do the translation from the preclinics to the clinics, and of course, medical doctors. So today we will talk about many different subjects, and we will start a little bit with the physics and chemistry of radioactive decay and radionuclides that can be used for cancer treatment. And then we will look at different types of radiation and why we can use certain type of radiation for treatment of cancer. And we will look at how radiation interacts with matter. And then we will look at several most famous examples and most successful examples of targeted cancer therapy. And we will also look at factors that influence the selection of a particular radionuclide for cancer treatment. So the majority of patients who have cancer and they have localized tumors, about 80% of patients can be cured by a combination of surgery or radiotherapy, which is external radiation therapy, and also chemotherapy. But there is a fraction of patients who are at late stage of the disease who will have distant metastasis, multifocal disease, which means that the disease is spread all over the body, and this fraction of patients need to have a systemic treatment such as targeted therapy. And this is where the radionuclide therapy comes into place. Ionizing radiation can actually be used in several ways for therapy, and one probably most famous and most widely used technique is called external beam radiation therapy. In this technique, the X-ray beam delivers a radiation dose to the tumor, and the patient is positioned on the bed. But the disadvantage of this technique is that it will damage not only tumors, but also healthy tissues that are on the way of the beam. A second option to use radiation for treatment is called brachytherapy, from a Greek word brachys, which means short. So it means that the radiation is delivered on a short distance from the source. And in this case, you see an image of a prostate gland, and there are a lot of small beads of small particles that are implanted in the prostate that will deliver the radiation dose to the prostate tissue. However, the disadvantage of this approach is that it is quite invasive. These beads are introduced surgically, so there can be complications from this kind of therapy. And the third type of therapy is a systemic radionuclide therapy, where the radiopharmaceuticals are administered systemically in the body. So the advantages of using radionuclides for treatment of cancer is that the main advantage is that development of resistance by cancer is very slow, especially to alpha emitters that have high LAT. And we will look later at what LAT is. Basically, the development of resistance to radionuclide treatment is very slow or non-existent. If we think about chemotherapy, usually cancer cells become very resistant to chemotherapy, especially after several cycles of treatment. They are able to adapt and find some alternative signaling pathways. But to radionuclide action, there is virtually no resistance because radionuclide kills cells by damaging DNA directly. Another advantage is that they provide very high efficiency of cell killing because they have this crossfire radiation. And you can see on this picture, it means that one radionuclide will emit a particle or a type of radiation that will be able to travel through several cell clusters. And because this particle will travel through several cell diameters, it will deposit its energy over several cells or even cell microclusters. That's why it's an advantage, because you don't need to target every individual cell, because in this case the action will travel through several cell diameters. And we will look, depending on the type of radionuclide, depending on the energy of the particle, this value might vary. There are some high energy particles and low energy particles. The third advantage is you can combine radionuclide therapy with other types of therapy in addition to other types of therapy, and you can possibly get enhancement or potentiation of the effect. And we also need to think that radionuclide treatment, it has a different toxicity profile than, for example, drugs or chemotherapy, because we need to look at different organs, especially organs that are most radiosensitive or sensitive to radiation, and these are particularly bone marrow and kidneys. So as you already know, radioactive decay is a process that happens when a nucleus breaks down into different particles, and then there is a release of energy. And there are several types of decay, and we will look at each type of decay. And first is an alpha decay. It's a decay with the release of an alpha particle. Alpha particle is basically nucleus of a helium atom, and it can be used for therapy. And the second type of radioactive decay is a beta decay. During this type of decay, there is a beta particle that is emitted, and it also can be used for therapy. Third type of decay is called gamma decay, or there is a release of a gamma ray. However, it's not very useful for therapy. It's used for imaging, for example, for SPECT imaging. And the fourth type of decay is positron emission. It's also not used for therapy, but it can be used for PET imaging. And just one note is that one radionuclide can have several types of emission at the same time. So when we consider each individual process, it doesn't mean that during a decay of a particular nuclide only that process happens. It can happen that during a decay there are several types of emission that are occurring, and sometimes it can be beneficial. For example, for lutetium-177, there is during the decay there is a release of a beta particle, so it can be used for therapy, but there is also some gamma that is released, and it can be used for imaging as well. So radiation emitted by nuclides can be in the form of an alpha particles, beta particles or gamma rays. And the energy that is released during the decay of an atom is can be quantified in joules or in the units of electron volts. So when radiation interacts with the matter, and we looked at different types of radiation, such as particles, photons, or neutrons, it causes ionization event. An ionization event means that there will be an ion formed, ion with one electron less. So radiation causes release of electrons from their electronic shells, and it's so-called ionization event. That's why we call radiation ionizing radiation. And there is a parameter that allows to evaluate the amount of energy that one particle emits during the decay, and this parameter called linear energy transfer. The linear energy transfer is the amount of energy that one particle emits per unit of track length, and that it deposits near its track length. So in this picture, you can see a track of an alpha particle. And alpha particles have a very high linear energy transfer. You can see that it's 5.3 mega electron volts. So they're very high LET, and you can look at the path of an alpha particle, and each black dot represents one ionization event. So you can see that there are a lot of black dots that are on the way of this particle. So it means that there are a lot of ionization events that happen along the track of an alpha particle. And we can look at the diameter of an alpha DNA helix. And we can see that an alpha particle has a very, very high chance to cause double strand breaks, just because these ionization events are very dense, they're very close to each other. So the probability of damaging a DNA is very high for an alpha particle. And on this picture, you can see a comparison between different types of radiation. And you can see for an alpha particle has a very high LET, so the probability of damaging DNA is much higher, so it deposits much more energy per micrometer of tissue than, for example, a beta particle. Beta particle only produces several ionization events and deposits a lower amount of energy per micrometer of tissue. And of course, the X-rays, they just go through the body and they don't deposit so much energy into the tissue. So there are types of particles that have high LET, and there are types that have low LET. And what we are interested in is causing a DNA damage, DNA breaks, and it is especially important to cause double strand breaks because they are more difficult to repair, and the probability to cause a double strand break for high LET particles is higher. So the double strand breaks are much more difficult to repair and takes more time. That's why alpha emitters are much more efficient at killing cancer cells than, for example, beta emitters. So what happens when there is a strand break? It can be a base damage, base of the DNA damage, and then it can also be a cross-linking between DNA and protein. And if there is a double strand break, the cells will go into mitotic catastrophe and then later in the apoptosis. So when we talk about radionuclide therapy, we consider three different parameters. One parameter is linear energy transfer. As I already explained, the second parameter is dose, which is energy deposition per mass unit, and the third parameter is dose rate, which is the dose per time. For example, dose delivered per minute, dose per hour, and the higher the dose rate, the more efficient will be therapy. And actually, for the external beam radiation therapy, when we have an external source, we can control the dose rate quite well. When we have targeted radionuclide therapy, when we administer the radiopharmaceutical, actually the dose rate is one of the most limiting factors to evaluate how efficient it will be, because of course using radiopharmaceuticals we consider circulation time in blood, and depending on the circulation time in blood, it is connected to the dose that will be delivered, so also connected to the dose rate. And there is also a secondary mechanism of damage during radionuclide therapy, and this mechanism is based on formation of reactive oxygen species. The reactive oxygen species are formed during ionization of water. So of course our cells have a lot of water, and also in the compartment that is in between the cells, intercellular compartment, there is a lot of molecules of water that can be ionized, and when they are ionized, they are transformed into radicals, such as hydroxy radicals or peroxides or superoxides that can damage DNA, and they can also damage proteins. And first we will just look at different types of radiation a bit more into detail, and we can start with the gamma radiation. So as I said before, gamma radiation just easily penetrates living bodies, so it's not depositing the energy, so much energy in the tissues. So the local dose is very low, and it also provides radiation of distant tissues. That's why it's not useful for radionuclide therapy, but it can be detected by external devices and can be used for a reconstruction of distribution of activity during SPECT imaging, for example. Alpha particles, as I said before, they have very high probability to cause double-strand breaks, and independently of the dose rate. And that's why they are very efficient, and they can deposit their energy up to several cell diameters. That's why they are suitable. They are more suitable for treatment of single cells and micrometastases. And also, they don't need oxygen to cause the reactive oxygen species through the secondary mechanism, because they are very efficient at killing cancer cells, so they destroy even hypoxic tumors. Sometimes there are some big tumors that have a hypoxic core inside the tumor, and they can be resistant to therapy, but in case of alpha particles, they kill even hypoxic tumors. The only disadvantage at the moment of using alpha particles is their availability. The availability of alpha emitters is currently very low, and the most commonly used right now are astatine, radium, actinium, and thorium. And on this slide you can see a summary of different nuclides and their characteristics. They have a different half-life. Here you can see the corresponding daughters that are formed during the decay, and if the half-life is very short in blood and there is a formation of daughters, it means that we are losing control over the parental nuclide. So after 1 hour we already have quite a large formation of some other nuclides with some other properties, which makes it rather complicated to evaluate, for example, safety. In the case of astatine and actinium, in the case of astatine it has a medium half-life, and in the case of actinium it has a very long half-life, over 10 days, and thorium has even longer half-life. And you can see that the availability is also different. So the only astatine is produced using a cyclotron, but all other nuclides are produced using generators. And in the recent 5 to 10 years, there was an increased interest in using alpha emitters, and currently more and more companies are interested in development of different ways to make alpha emitters more available. And now we can talk about the beta particles. So as I mentioned before, beta particles, they have slightly lower LAT than alpha particles, and we also need to consider a parameter which is called an energy of a beta particle. So there are some nuclides that decay and they emit high energy particles, such as yttrium-90 emits a high energy beta particle, and iodine-131, it emits a low energy beta particle. So why is it so important? Because that means that the energy will be deposited in a different, over a different diameter in the tissue, and we can look at the comparison between yttrium-90 and iodine-131, and we can see that the absorbed fraction for high energy emitter will be low at sphere masses of a low mass. Why is it the case? Because the energy will just travel outside the sphere mass and will be deposited mainly at the rim of the sphere. So it will just go through the sphere and mainly be deposited outside. That's why for low masses, the absorbed fraction for yttrium will be quite low. But if we look at iodine-131, we can see that the absorbed fraction is very high, both for small spheres and for large spheres, because the energy of the beta particle is small, and it will not travel so far in the tissue from the source of decay, and that means that the energy will be deposited equally well in small spheres and in large spheres. So how is it translated into living organisms and tumors? Well, it means that for every tumor size, there is an optimal optimal value of absorbed energy. So for example, we can look at the comparison between astatine, which is an alpha emitter, yttrium, which is a high energy beta emitter, and lutetium or iodine, which have lower energy of beta particles. And for astatine, it's alpha emitter, so we can see that already at very small sizes of tumor mass, the absorbed energy is very high. And maybe you remember that I mentioned alpha emitters can be used for treatment of even single cells and micro clusters of cells, because the absorbed energy is already very high at very low tumor mass. But if we look at yttrium-90, which is a high energy beta emitter in green, we will see that the absorbed energy increases almost linear with the mass of the tumor, and it reaches maximum only for tumors of a very high mass of about 1 gram. But if we look at some lower energy beta emitters such as iodine or lutetium, they already reach maximum absorbed energy at a mass which is slightly lower, so a mass of about 1 milligram. So different nuclides are optimal for different tumor sizes, and for low energy, for low LAT emitters, the effect depends on the dose rate. Also, effect depends on hypoxia and oxygenation of tumors, because sometimes the energy of the beta particle is not enough to kill, to cause DNA damage on itself. That's why you need to have a secondary mechanism of damage, reactive oxygen species. And in comparison for gamma, the dose is more localized for beta particles. That's why they can be used for treatment. Currently, there are 4 radionuclides that are commercially available that are beta emitters. It's rhenium-188, yttrium-90, lutetium-177, and iodine-131. They all have a different half-life. Also, we need to think about the connection between half-life of the radionuclide, the circulation time in blood, and for how long the nuclide will stay in the body, so the pharmacokinetics and the dose that will be delivered to the patient. So the half-life will have a direct influence on the dosimetry profile also. And then the different nuclides have, they emit particles, beta particles of different energy. There are high beta emitters, high energy beta particles that are emitted from rhenium and yttrium. So they travel much further into the tissue. So the range in the tissue is much higher than for emitters with the low energy beta particles. So they will travel less into the tissue, and they are more suitable for treatment of small sized tumors, while those ones are suitable for treatment of large tumors. And there is also a parameter of irradiation of photons. If there is a photon radiation, then it means that we can monitor the treatment by doing SPECT imaging. And in case of lutetium or iodine, we can administer radiopharmaceutical for treatment, but we can also image a patient, and by doing imaging, we can calculate the dose to the organ, make an estimation. Of course, using SPECT imaging will not be as precise as using PET imaging, but it will still provide some important information. For example, if radiopharmaceutical accumulates in the tumor or not. And in general, if we talk about targeted cancer therapy, we can divide it into 2 strategies. One strategy will be passive targeting. It means that the radiopharmaceutical would just be accumulate in the organ or in the tissue of interest on itself without any active mechanism of uptake. So in case of passive targeting, we are taking advantage of some native chemical properties of a nuclide, and taking advantage of the fact that it can participate in different biochemical processes in vivo. For example, for iodine-131, we know that its iodide is taken up by sodium iodide symporters, and these symporters are expressed in thyroid gland or in the stomach, for example. And because it's expressed in the thyroid gland, it can be used for treatment of thyroid cancer. And therefore, a second example is radium-223. So radium is in the same group in the periodic table as calcium. That's why radium is a calcium analog. It will accumulate in the bones, so it can be used for treatment of bone metastasis. And then we have a second type of targeting mechanism, which is active targeting. So in active targeting, there is a process of molecular recognition of target on the surface of tumor cell. For example, it can be a ligand receptor interaction through some protein-protein interaction, like we talked about quite a lot in this course. So a few examples of passive targeting. So as I mentioned, in passive targeting, we are relying on metabolism and accumulation of a particular nuclide in the body. And one of the oldest, the most well-known examples that is used over 60 years in clinics is iodide-131, because iodide accumulates in the thyroid through the uptake by sodium iodide symporter. and it can be used for treatment of thyroid disorders such as hyperthyroidism and thyroid cancer. The disadvantage of this method is that iodine 131 has an unfavorable radiation profile. If you remember from this table, iodine 131 has quite a high energy of photons, which means that they will travel through the tissues and outside the body. They will deliver quite a large dose to the patient and can even deliver a dose to the personnel or to people around the patient. So its use is limited. It's still used, but it's limited nowadays. And just a question. So after Chernobyl's disaster, people were given a legal solution, which is 10% potassium iodide and 5% iodide. Do you know why? What was the purpose? Maybe someone has an idea.
Obviously, to substitute radioactive iodine to non-radioactive iodine in this solution.
Sorry, in the beginning I missed. Can you repeat once again?
Iodine solution was taken for substitute radioactive iodine by non-radioactive.
So actually, I don't think it can substitute. It's the point more here is to prevent the uptake. So to saturate the transporter. So it will be most efficient if you take it before the iodine is taken up, because if you are able to take iodine as early as possible, you will saturate the transporters, and the radioactive iodine would not be able to accumulate. But if it's already in the body, if the radioactive iodine is already in the body, it would accumulate. So it will be a bit less efficient. But yes, the general logic is that if we can take it as early as possible, then we will be able to compete out the cold, because it will be in a large access to the radio-labeled iodide. So sometimes in our experiments, in mice, we give mice water that has potassium iodide in it, but we give it a few days before the experiment so that there will be time for this process to happen. So usually a few days is sufficient to saturate the uptake of iodide in the thyroid. And the second example of passive targeting can be various calcium analogs, as I said before. So we have strontium 89, radium 223, and samarium 153, which are all calcium analogs and can accumulate in bones. So for example, strontium 89 is a high energy beta emitter, and the pharmaceutical name is metastron, which was approved by FDA for treatment of bone metastasis. And then later there was a second radiopharmaceutical, which is called alpha-radin, which is basically a chloride form of radium 223. So it's just radium chloride. And radium is an alpha emitter with a very high LAT, which is also used for treatment of bone metastasis. And when it was first developed and tried in patients, it was so effective that it got approved by FDA using so-called fast track, which is only for radiopharmaceutical that have shown a great efficiency in clinical trials. So at the moment, radium 223 is very widely used for treatment of bone metastasis or patients with bone metastasis. Now we can talk a little bit about targeted cancer therapy. So in the targeted cancer therapy, there is this concept of having a magic bullet, which is not new. It was introduced a long time ago, which is the point is that we will be able to selectively target a pathogen in the body or cancer cells in the body by using this construct that consists of two parts. In one part, we will have cancer targeting molecule. Let's say it can be a protein, it can be an antibody. There were also some examples of different carriers. It can be a nanoparticle carrier if it will selectively accumulate in a particular tissue. And then we will have a therapeutic part connected to the first part by some kind of linker, covalent bond. And in the therapeutic part, we will have some drug molecules, we can have toxins, or we can have radionuclides. And if we think about different targets for cancer therapy, if you would like to know more about it, you can look in these two papers from 2000 and 2011 about cancer biology. They basically summarize all the hallmarks of cancer, all the characteristics of cancer. We can take one particular characteristic and we will find some kind of upregulation of a molecule, for example, overexpression of a receptor on the cell surface, and then it can be used as a target for cancer therapy. And here are the three most common examples of different targets in cancer therapy. For example, for treatment of lymphoma, the target is called CD20, and its function is to mediate immune response of beta cells. And there are two monoclonal antibodies that are radiolabeled. One is called Zevalin, another one is called Bexar. So they target CD20 on the surface of lymphoma cells. Second type of target is somatostatin receptor. So these receptors are overexpressed in neuroendocrine tumors. And to target these receptors, we can use somatostatin analogs. Maybe you have heard about dotatate or dotatalk. So these analogs will target somatostatin receptors. And in case of prostate cancer, one well-known target is PSMA. That is overexpressed on the membrane, and there are different targeting molecules, but one that is approved right now for imaging is called the prostacin. And we will look now a bit more into detail at every type of every type of cancer, every type of target. So what we need to think about when we develop a radiopharmaceutical, we need to think about the pharmacokinetic profile, because it will be directly related to the dosimetry profile, or to which dose will be delivered to the tumor and the dose that will be delivered to normal organs. So we need to think about the absorption rate. Of course, most radiopharmaceuticals are administered IV, so they go directly in the bloodstream, so the absorption will be 100% in this case. Then the distribution, how it will be distributed in the body, where it will go, are there any transporters that are involved, is it an active mechanism of uptake or passive mechanism of uptake. Then we need to think about metabolism, in which organs is it broken down and excreted. Is it taken up in liver or in kidneys? And excretion, if it's more lipophilic, it will accumulate in the liver and in the intestine, and will leave with the fecal masses. But if it's more hydrophilic, then it will be excreted through renal pathway, through kidneys, and leave the body through urine. And we really need to think about different factors that can affect the selection of a nuclide. We also call it label. And the factors are the tumor size, as I explained before, cellular processing of a protein by cancer cells, because it will affect retention of a nuclide in cancer cells and in normal cells. And we talked about it quite a lot in this course, but I will also explain it once again. And the third factor is uptake of protein in excretory organs, and then the size of the targeting protein, because the size will influence the half-life in blood, and the half-life in blood means also how well it will accumulate in the tumor and how fast it will be excreted from the body. So I suggest that we can make a 5-minute break right now, and after 5 minutes we can continue with the second part. Okay, so we can continue. One of the important properties that we should evaluate is internalization of radiolabeled proteins. Depending on which kind of target that you are studying, which type of receptor it is, and how it behaves after binding of the protein to the target, depending on the internalization, how fast it occurs, you might be able to say which type of label will be better, if it will be radiohalogen or radiometal. And we looked at several kind of radionuclides can be used for treatment of cancer treatment, and out of them radiohalogen is for iodine 131, and the majority is radiometal, so they would have more residualizing properties. And after internalization, the protein will go inside the cell and will be inside the organelles that are called lysosomes. And inside the lysosomes, we have a very acidic environment and a lot of different enzymes that can degrade the radiolabeled protein. And there can be a formation of radioactive catabolites. Depending on their property, they will be either lipophilic, and so they will diffuse outside of the cell, or hydrophilic, they can be retained inside the cell. So the residualizing labels will be mostly radiometals, and non-residualizing will be radiohalogens. And on this graph, you will see an example of a retention of various nuclides by cells, by cancer cells. So it's trastuzumab monoclonal antibody that was labeled with lutetium or with iodine, and you can see the retention of activity in cells is very different. Why? Because trastuzumab is very fast internalized, so basically we are losing the label from the membrane. The label just goes inside and outside. But in case of lutetium-177, the antibody is internalized, but the label stays inside the cell. So we are not losing the label in this case. Probably you have seen this picture before. It just shows the retention in excretory organs. So as I said on the previous lecture, I think I said that in internalization in excretory organs is very fast. That's why we see such a difference between liver in the case of iodine and liver in the case of radiometal or indium. Because internalization is very fast, the activity disappears from excretory organs because iodine is not residualizing label. But internalization is, if it's fast and we have a metal, the activity will just stay in excretory organs. So it is not maybe so important for imaging, because we are just doing, for example, single imaging, and we are not delivering so much dose to the patient. But for radionuclide therapy, it becomes very, very important, because usually radionuclide therapy is given at a very high dose, a very high activity that we inject, and it's usually several cycles of treatment. So will be repeated damage to normal tissues, and we need to make sure that we do not exceed a safety level to normal tissues. And the size of the targeting protein is also quite important. I also showed this scheme in one of the previous lectures. The smaller the size of the targeting protein, the better it will go inside the tissue, and the faster it will be cleared from blood. So there is a 60 kilodalton value, which is a kidney barrier, kidney filtration barrier in the glomerulus. So molecules that are more than 60 kilodalton, they will be circulating in blood for a long period of time, usually several weeks. And molecules that are small, below 60 kilodalton, they will be excreted from blood much faster. So originally, monoclonal antibodies were the first molecules that were used for radionuclide therapy, and this approach is called radioimmunotherapy. So if you will search literature, you will see this term radioimmunotherapy. It means using monoclonal antibodies, so immunotherapy, and radio means radionuclide therapy. So this term is related to the using monoclonal antibodies. So historically, they were the first targeting vectors, even starting from the 1990s. They were used for radionuclide therapy. They have high affinity to targets. And the advantage is that we can just take a therapeutic antibody that is already approved for use in clinic, and we can radio-label it. So it seems like a very easy solution, but it's not an optimal solution, because antibodies have a very long half-life in blood, so it will just circulate in blood for a long period of time and deposit a lot of radiation dose to normal tissues, and especially to bone marrow that is very radiosensitive. However, there were some clinical success using radioimmunotherapy, so monoclonal antibodies for treatment of lymphoma. And if you're not familiar with biotechnology, you can, if you see a name of the molecule and you see MAB at the end of the word, it means it's a monoclonal antibody. So in this case, it's ibrutinib and tezepelumab. So it's two monoclonal antibodies. In this case, it was labeled with yttrium-90, and in the case of Bexar, it was iodine-131. So they were developed in the early 2000s, and they were tried. They were compared in clinical trials with the conventional treatment. So in this case, it was just a cold antibody, and in this case, it was cold chemotherapy. So you can see in red are the numbers of overall response rate or complete response rate. They were much higher for patients who were treated with the radioimmunotherapy compared to the conventional standard treatment. So these were two very successful examples for treatment of lymphoma, but actually for Bexar, it was withdrawn from the market, but not because it was not effective, but because it was a commercial failure. So it was not effective. It was not related to the efficacy of treatment, but due to some other reasons. And then people started to look into proteins of different sizes, mostly antibody fragments. And as I said before, antibodies were historically the first molecules, but because they have a very long half-life in blood, they might not be optimal. And people started to look at fragments of a much smaller size, where they can genetically engineer various fragments of a smaller size. And here you can see an example of a study where they engineered size of about 75 kilodalton. So it would still not go through the filtration barrier, so it would still have some prolonged half-life. And then another fragment of a size below 60 kilodalton, so it's 50 kilodalton, so it will go through the barrier into the urine and will be excreted faster. So in this paper, they have looked at blood clearance over time, and they have looked at accumulation in the tumor over time. And then when comparing blood clearance and accumulation in the tumor, they found that the medium size fragment, which was a 75 kilodalton fragment here, had the most optimal properties. So it had high accumulation in the tumor and a medium optimal half-life in blood. So they have chosen this molecule, and they used iodine-131 as a therapeutic nuclide, and then they compared survival in mice that had tumors implanted. So they had three groups of mice. One was the control injected with the saline solution, and two treatment groups. One of them was injected with 37 megabecquerel and another one with 74, so twice higher dose. And it was not very surprising that they saw the longest survival in the group of mice that received twice higher dose of treatment compared to the control also. So this was the first indication that radioimmunotherapy is efficient in case of lymphoma, but there are several problems, and especially for treatment of solid tumors. We need to think that solid tumors are very different from lymphomas because they have high radio resistance, so they are more difficult to treat with the radiotherapy. Another disadvantage is the low dose rate that we deliver during this kind of therapy, and also low dose delivered to the tumor, because antibodies are quite large molecules. They do not go too deep inside the tumor tissue. And that's why it's slow extravasation and slow penetration in the tumor is a limiting factor. And another factor is slow blood clearance. So they are cleared from the blood very slowly and deliver high dose to the bone marrow. And then researchers started to think about different solutions, and one indication could be reduction of the size of targeting protein, as I mentioned on the previous slide. Reduction might be beneficial, and in next examples you will see some successful examples of using short peptides which have a very short half-life in the body, but they are quite efficient for treatment of different kinds of cancer. So the first example, maybe you have heard about somatostatin analogs such as Dotatalk or Dotatate. Because I don't know so much your background, maybe you can, if anyone has heard about this kind of treatment, can you just indicate if you heard about it?
No, don't have.
So I guess no one heard about it.
Obviously, yes.
Sorry, the sound is very low. I don't hear so well.
Obviously, yes. It looks like nobody heard about.
Okay. So then I will just quickly explain about this concept and about neuroendocrine tumors. So neuroendocrine tumors are a fraction of tumors that originate mostly in the gut area. The primary tumors mostly originate in small intestine, some pancreas, and a large fraction in lungs. And usually the metastases are most commonly found in liver, lungs, and bones. So in this example, you can see a widespread of metastases all over the body, and in this case, the patient received a PET scan with a gallium labeled Dotatalk, which is an analog of a somatostatin. And somatostatin is a short peptide consisting of 14 amino acids, and it has a disulfide bridge connecting the two cysteines together. Later, the structure of somatostatin was modified, and they have decided to reduce the size from 14 to 8 amino acids, and they have identified some crucial amino acids that are important for binding. So the analog is called octreotide, and it was conjugated to a chelator called DTPA, which I have mentioned during one of the lectures. It's acyclic chelator, can be used for labeling of radiometals, for attachment of radiometals to the molecule. So that was the first generation, and this molecule can be found under name octreoscan. So indium labeled DTPA octreotide can be used for imaging, and it's called octreoscan. But the same analog that has a cyclic chelator, such as DOTA, for example, dotatate, can be radiolabeled with lutetium-177 and used for therapy. And just on the short slide, on this short video, I think it's a very nice short video that can illustrate this concept quite well. So I would just like to show it to you. So basically, here we see a molecule which is an octreotide, which has a chelator and which has a lutetium inside, and lutetium is a beta emitter, so it emits a beta particle, and also so it's therapeutic part, and also it emits a gamma ray, and the gamma can be detected by SPECT. So during the decay of lutetium, there is a beta particle that is emitted, and the radiopharmaceutical is injected into the bloodstream, so it's is distributed all over the body. Because it's a very short peptide, it has a very short half-life, and when it reaches the tumor, it is delivered to the tumor, and because it has a very small size, it is able to penetrate in the tumor tissue quite well and bind to the receptors. In this case, we see an internalization of the receptor inside the cell, and because lutetium is a residualizing label, its catabolites are also retained inside the cell, and after damaging of DNA, there is an event of tumor apoptosis, so programmed cell death and the reduction in the tumor size. And as I said before, for lutetium, we can also monitor the delivery to the tumor by SPECT. In this case, it's 3 days after injection imaging. Because lutetium has quite a long half-life, it allows to do imaging several days after injection. So lutetium dotatate was approved by FDA in January 2018 for treatment of neuroendocrine tumors because it showed to be quite efficient at delivering delivering the dose, at the same time not delivering too high dose to the normal organs, so also being quite safe. And if we look at this graph, we can see the proportion of patients who were treated with the lutetium dotatate and who got some kind of response. So in green, it's positive response. We can see that it's a complete response, partial response, or remission. So almost 50% of patients who were treated with lutetium dotatate got a benefit of treatment, and also 34% got stable disease, so there was no further progression, and only 20% of patients had progressive disease. So in 80% of patients, it was quite effective. So these trials were done about 10 years ago, and the first message that the clinicians got is that if you reduce the size of a targeting protein from monoclonal antibody to a very short peptide, it might be more efficient, so it might be the key to success. And after seeing these results, researchers started to look more into short peptides or small targeting molecules. And the second example of targeted radionuclide therapy is prostate cancer therapy. In prostate cancer, the target is PSMA. PSMA receptor is a prostate-specific membrane antigen receptor, which is overexpressed in a lot of prostate cancer patients. And the ligand to this target is usually small molecule that is below 5 kilodalton, and in this picture it's a ligand called PSMA 617, but actually it's can be a lot of different ligands, and this field is very actively developing, and every year there are new ligands that target PSMA that are on the market. So the theranostic pair that is used currently, one of the pairs is using gallium for imaging with PET and using lutetium for a treatment. So when we label the same molecule with gallium, we can do quantification in different organs because it's PET, and using lutetium, we can use it for therapy. So in this case, can see a patient that was imaged before treatment, and then it received several cycles of treatment, and you can see the disappearance of metastases except one. So after several cycles of treatment, after almost 6 months of this kind of therapy, it was quite effective. And this is a very interesting example. It's quite unconventional and not very standard, but it can really illustrate the difference between beta emitters and alpha emitters. And on this slide, you can see a patient who got gallium PET scan with PSMA. So patient had a lot of metastases in the gut region, and then it was given lutetium as a treatment, 2 cycles. But after 3 months, they saw that it only became worse, so the treatment was not efficient at all. And then they given the same patient actinium-225, which is an alpha emitter. So after 2 cycles of actinium-225, we see a complete disappearance of majority of metastases in the gut area, and also after an additional cycle of treatment on the disappearance of some residual metastases. So in this slide, you really see that alpha emitters are very, very powerful at killing cancer cells compared to beta emitters. One thing that we need to think about is we see accumulation in salivary glands here and also in kidneys. So PSMA has natural level of expression in salivary glands and kidneys, and one big side effect from this kind of treatment is patients don't have any excretion of saliva after this treatment, or they have very dry eyes. So that is quite a severe side effect, but also they can be damaging effects to kidneys. So many people are very positive about this kind of treatment, but we need to think about side effects, and sometimes side effects can be quite bad. And actually, at the moment, it is not known how dangerous are these side effects, how dangerous is accumulation in kidneys, and because it was just published in 2016, and during the last 3 and 4 years, people are learning more about this kind of treatment, learning what we can give to the patient and what is the tolerance level, what is the safety level. So this treatment is not approved yet, but there are hundreds of clinical trials all over the world using PSMA therapy for prostate cancer. So another important message is we need to use personalized treatment, which means that the treatment should be adjusted specifically to a single patient as much as possible, because earlier the treatment was given as a standard, for example, a single infusion at a dose level based on some other findings, or a single dose was administered to many patients. But if we just giving a single dose which will be a maximum tolerated dose. This can be activity determined for the least resistant patients, so there is a risk that we will not treat sufficiently many patients, so there will be undertreatment of patients, and we need to develop patient-specific dosimetry for every patient. And for example, for lutetium dotatate, there were this kind of studies where they administer different cycles of lutetium dotatate. One cycle is 7.4 gigabecquerel, and then they evaluate the dose to the bone marrow and the dose to the kidney. And then they look at a number of cycles which a patient receives before reaching a critical value to the bone marrow and to the kidney, so that they could see that in totally over 200 patients, the majority reaches the critical level after 4 cycles. But there are some patients who can tolerate much more cycles, 5, 6, 7, and 8 cycles before reaching this safety level. And when they compared the survival of patients who got treatment based on this kind of data, so in red you can see patients who received a certain number of cycles with a dose less than 23 gray to kidney. Their survival is much less compared to patients who actually got more cycles, who got this critical dose to the kidney, but they also were surviving longer. So this is an indication that first of all, dosimetry should be used to evaluate the safety of treatment, and the second is that perhaps the current limitations, the current limits of 2 gray to bone marrow and 23 gray to kidneys can be overcome and can be perhaps even higher, because these numbers were these numbers came from I think external radiation treatment, and in the case of radiopharmaceutical treatment we might have a different scenario. So this data shows that actually the more activity we give to the patient, the longer they will survive, so that might be beneficial. So in the future, we need this kind of studies will be very necessary to really evaluate, should we be scared of radionuclide therapy, or we can actually exceed the safety limit because it will prolong the survival, prolong the life of patients. And another approach that is also quite interesting, you can think about combining several radionuclides, because we not only have, usually in patients, not only have tumors of small size or only of large size. They can be a mixture of large and small tumors. And in order to treat them efficiently, we can apply a mixture of, for example, high energy beta emitters such as yttrium and low energy beta emitters such as lutetium. And this was first tried in mice that had xenografts. And in this case, they saw that the combination of lutetium and yttrium gave the longest survival compared to the control group, or just nuclides on themselves alone. And actually, I didn't look at the statistics in Russia, but if we look at the statistics of therapy in Sweden, we can see from 1994 to 2018 that the majority of cases is iron 131 for the treatment of thyroid cancer. Then we have strontium 89 and radium 223, which are used for treatment of bone metastasis. We can see that for radium since 2002, basically since the FDA approval, its number is growing quite a lot because it's such an efficient treatment. And then we can see that example of neuroendocrine tumors, indium and lutetium octreottate So lutetium dotatate. Also, the number of cases that, the number of patients that receive this kind of treatment is also increasing, especially after FDA approval. It also increased quite a lot. And if we look at the diagram, the total proportion of radionuclide therapy is still passive targeting. It's not an active targeted therapy, so it's still radium-223. And there is only a small fraction of targeted therapy. So the take-home messages from this part is that radionuclide targeting provides several advantages. So it's there is virtually no resistance to this kind of cancer, to this kind of treatment. The radionuclides provide crossfire radiation, and also there is no alternative signaling pathway, so the less chances of cancer cells to develop resistance. We also need to think about toxicity profile, which is different for nuclides than for drugs. And this kind of treatment is also independent of immune system of a patient. Then there are several factors that we should consider when we choose a particular nuclide. It's size of the tumor, size of the targeting protein, cellular processing of protein by cancer cells, and uptake of protein in excretory organs. So the radioimmunotherapy using monoclonal antibodies was successful for treatment of lymphoma, but not successful for treatment of solid tumors. And the possible solution can be just to use it for treatment of minimal disease or small-sized tumors. Otherwise, we need to reduce the size of radiolabeled protein from monoclonal antibody to a short peptide, because reducing the size, we can reduce the exposure of bone marrow and also improve extravasation and delivery of radioactivity to the tumor. We also need to think about personalized treatment and dosimetry that will be specific for every patient. And of course, we need to develop some novel targets, novel tracers for other types of cancer that would have the most optimal pharmacokinetic profile. So just one thing that I would like to mention is that radionuclide therapy at the moment is not used to completely remove the disease and to completely cure the disease, but it's rather aims to extend survival in combination with other options, and it's mostly used to provide palliative care, especially radium-223 treatment for bone metastases, mostly used to reduce the pain and reduce some symptoms, and of course to improve quality of patient's life. But as we saw during this lecture, there are some alpha emitters such as PSMA in combination with alpha that has shown complete cure, but it's still quite early to say that it will be used to completely eradicate the disease. So at the moment, its aim is to extend the survival. And right now, I would like to show you some research that we are working in the laboratory of Professor Tolmachev here in Uppsala. We're using engineered scaffold proteins for therapy. And as you probably know, we are working with Affibody molecules. So Affibodies are small proteins of about 6 kilodalton. They are much smaller than monoclonal antibodies, and they have very high affinity to the target. They are robust, so they can be radiolabeled at the various pH. It can be heated mostly to 90-95 degrees. They're also quite easily produced, can be produced recombinantly or synthesized using a peptide synthesizer. And also the labeling can be site-specific at a certain position. If we introduce a single cysteine, we can attach one label or one radionuclide per one molecule of an antibody. And mainly we're working with HER2, which is a human epidermal growth factor receptor. So HER2 is overexpressed in breast cancer, also gastroesophageal cancer and ovarian cancers. And on this case, this slide, you see a comparison between trastuzumab, which is a monoclonal antibody, and an Affibody. So Affibody works very well for imaging. You can see quite a lot of well-imaged metastases, but Affibodies cannot be used for therapy because they accumulate in the kidneys to a very, very high degree. Almost 90–95% of Affibodies that are injected in patients accumulate in kidneys. So this is a challenge for therapy, high kidney uptake, and it's much higher compared to the uptake in the tumor. So in order to overcome this problem, there were several strategies. And so the first strategy that was developed is we can optimize the properties of the label and the Affibody, so we can optimize lipophilicity profile. And the first work was done in 2007. You can see in this case there is some accumulation in the tumor, but there is also accumulation in the cecum, so it was very lipophilic molecule. And then they have changed 3 amino acids in the scaffold, exchange glycine to glutamate. And by exchanging 3 amino acids, the pharmacokinetic was changed to renal excretion, and in this case we can see accumulation in the kidneys. And then there were done some further optimization to reduce the accumulation in the kidneys, and here at the final image you will see that the accumulation in the tumor is very high, but accumulation in kidneys is much lower. So this could work very well with technetium for imaging and the theranostic pair with rhenium, for example, for therapy. The second strategy that was developed is called pre-targeting strategy. Because this strategy cannot be used for all radionuclides, the pre-targeting strategy can be used for radiometals, for example, lutetium-177. And in this pre-targeting strategy, we are splitting the molecule into 2 parts. One will be targeting the cancer cells, and the second one will carry the radioactivity. And each of these parts will have a recognition tag on the surface. So they will be able to find each other in the body and react with each other. And on this slide, you can see the general scheme, how it works. So first we administer primary agent, which is an Affibody with the recognition tag. And after it's bound to the tumor cell, we can administer secondary agent, which is radioactive. And then they will be able to react with each other at the tumor site. And as a recognition tag, so as this red and blue on the scheme, we are using PNA, which is a peptide nucleic acid, which is an analog of a DNA, but it's much more stable. It's not degraded in vivo. It's not degraded by nucleases. So it's very stable and has a very high it's very efficient hybridization between the 2 strands. And here you can just see an example of 3 groups of mice. In the first group of mice, we administer an Affibody that was radiolabeled directly with indium. So of course we see high uptake in the kidneys and some accumulation in the tumor. In the second part, we have a pre-targeting. So we first administer a primary agent, then we wait for 16 hours. It's cleared from blood and it's also removed from the kidneys. Do you remember the internalization in normal organs is very, very fast. So this primary agent is removed from the surface of liver and from the surface of kidneys. and we wait for 16 hours for this process to happen. After 16 hours, we administer radioactive secondary agent, which only binds to where the primary agent is on the cell surface. So because the internalization on the cell surface of tumors is very, very slow, this reaction is very efficient. And after 16 hours, we can catch the primary agent at the tumor and deliver the radioactivity. And in the case of the third group of mice, which was used as a negative control, it only received the secondary radiolabeled agent without the primary antibody agent. So we can see that the secondary agent has almost no accumulation in the body except some low uptake in kidneys. So this kind of therapy was used in mice that had HER2 expressing xenografts. And you can see on this slide is the survival of mice who received, in black is the control group who received saline, in green is just primary agent alone, blue is just radioactive secondary agent. So all of these groups died very early from cancer, but the group that received a combination therapy of both primary and secondary had twice longer survival. So 6 cycles of this therapy with lutetium-177 improved survival of mice. And in this case, in another study, another strategy was to prevent the excretion of an antibody through kidneys by fusing antibody with an albumin binder. So in this case, an antibody would circulate in the blood for a long period of time and would not be taken up by kidneys. And in this case, we can see that mice who were treated with targeted treatment 21 megabecquerel, 17 megabecquerel, they survived quite long in comparison to mice that received PBS or unspecific. And then I think I also showed this example before, why mice died. In this group, they also received treatment, but they received 30 megabecquerel. And in this case, they died very early on because this dose caused toxicity to bone marrow and depletion of red blood cells. And that led to early death. So this dose was too high. So this kind of approach shows that we can use antibody molecules labeled with lutetium connected to this ABD, which is an albumin binder, to treat cancer in this case. Yes, so this was everything that I wanted to say for today's lecture. If you have some questions, we can discuss. You can also ask them in Russian or type them in the chat because the sound seems to be low.
Thank you for lecture.
Thank you very much. As usual, there will be the record of the lecture and also the slides will be available.
Спасибо.
Все, спасибо большое.
Спасибо за лекцию. Очень
Да,
интересно было.
спасибо. Хорошо, хорошего дня тогда. До свидания.
До свидания.